Let's just jump right in. Today, we're exposing the one tiny mistake that silently costs Cambridge A-level biology students their absolute best marks. If you've been struggling to secure those top band scores in the 9700 syllabus, specifically when we're talking about chromosome behavior during mitosis, well, you are in exactly the right place.
So, look at the question right here on the screen. Do you know the #1 mistake costing students their marks in mitosis? Think about how much time you spend studying prophase, metaphase, anaphase, and telophase. You've probably got all the diagrams memorized, right? But what if I told you that simple memorization is exactly what puts your grade boundary at risk? You see, the exam doesn't just test your memory, it tests your analytical depth. One single structural error in how you write your answers can completely mask your otherwise perfect biological knowledge.
And here it is on the slide. The #1 mistake is confusing cause and effect in chromosome behavior. You literally can't just write chromosomes move to the poles and expect full marks. You have to explain the why. The fact that the shortening of the spindle fibers actually causes the sister chromatids to separate. If you're just listing facts without linking the mechanism to the outcome, well, you're going to hit a wall in the mark scheme.
Now, what's really fascinating about this quote on your screen is that it comes directly from the principal examiner reports. Candidates consistently confuse key terminology. Always define terms before using them in extended responses. This isn't just a helpful suggestion from me. This proves that academic benchmarks literally demand precise definitions. The examiners themselves are warning us. They're telling you point blank the top scoring answers require you to actually define your terms before launching into an extended response. So, to keep you out of those traps, let's run through the top 3 exam mistakes in mitosis. First, as we just talked about, confusing cause and effect. Second, forgetting to check your units and define your terminology. And third, providing answers that aren't specific to the given context. If you just write out a generic textbook list you memorized, instead of applying it to the specific cell context given in the exam, it's just not going to score highly.
Now the examiner clue. In a moment, I'm going to show you this exact hidden grading parameter that completely transforms a standard answer into a top band response. It's an absolute game changer for handling those complex data and graph questions. But first, you kind of need to understand the framework that makes that clue obvious. Because if we don't have that foundation, the clue just isn't going to make sense. The method mark strategy. Let's look at this procedural framework that honestly changes everything. Okay, here are the steps. This is your standardized sequence for tackling complex problems. You don't just blindly jump to the answer. Step 1, identify the key elements. Figure out what the stimulus is actually showing you. Step 2, apply the relevant biological principles. Step 3, execute your analysis with really clear working. Step 4, verify your results and make sure your reasoning makes sense. And Step 5, present it all in an appropriate format using exact Cambridge terminology. Now, let's apply that strategy to the chromosome behavior stages, prophase, metaphase, anaphase, and telophase. When examining these stages chronologically, it's not just about drawing what chromosomes look like at a specific minute. It's about processing the data. You need to identify the key elements of the phase, apply your cell division principles, and verify your results against the exam's timeline.
And this brilliantly illustrates our core principle for today. The assessment framework prioritizes analytical depth over superficial breadth. Simply naming prophase or anaphase, that's superficial. But explaining the causal relationship, like how spindle fibers contracting separate sister chromatids, and backing that up with the data provided in the specific exam context, now that is analytical depth.
The interactive challenge. Spot the error. Let's see how this all builds by looking at an actual Cambridge-style example. Look at this visual comparison on the screen. On the left, we have a flawed student answer. It's how most students answer, honestly, like a jumbled pile of puzzle pieces, just throwing generic mitosis terms onto the page with no logical flow. But on the right, we have a top-band answer. Look at that structure. They take the biological concept, snap it into the evidence or data provided, connect it with scientific reasoning, and lock the whole thing in with precise examiner wording. They connect the dots perfectly. Pause and predict. Where did the flawed answer fail to check for reasonableness? Seriously, I want you to pause this explainer right now and think about your last past paper attempt. Can you pinpoint exactly where you might have failed to justify your biological decisions with evidence? Think about how snapping those puzzle pieces together structurally would have saved your grade. Evaluating the evidence. Now that we've seen the answer structure, we need to talk about handling data interpretation. Because part of evaluating evidence is knowing exactly which statistical test to use on your exam data. Look at this table.
If you're comparing means, you use a t-test. If you're dealing with categorical data, like, say, counting the number of cells in prophase versus anaphase, it's chi-squared. And if you're looking at relationships between variables, you use correlation tests. Selecting the correct statistical tool is an absolute non-negotiable step in your method.
Remember that examiner clue I promised earlier? Well, here it is. The secret to top band marks is high-scoring critical analysis. The formula is deconstruct, solve, and evaluate. You can't just solve the problem, you have to evaluate the biological context. You need to identify given information, break the problem into logical steps, and question assumptions. Consider different perspectives and identify limitations and their effect on conclusions. If your answer just spits out a calculation without questioning the assumptions made in the data, you are literally leaving marks on the table.
This quote on the screen perfectly reinforces our examiner clue. Top-scoring candidates demonstrate evaluative thinking by questioning assumptions, considering different perspectives, and justifying decisions with evidence. Evaluative thinking is exactly what bridges the gap from a standard, good answer to an elite, top-band answer. Whenever you write an extended response on chromosome behavior, ask yourself, have I actually justified my decisions with evidence? Have I considered the limitations of the data?
The binge loop conclusion. Let's quickly recap what we've solved today before looking at the final missing piece of the puzzle.
Here are 3 key takeaways to immediately protect your grade boundaries. Number 1, never confuse cause and effect in mitosis stages. Understand the why, not just the what. Number 2, relentlessly use the deconstruct, solve, evaluate mark strategy to ensure you actually get marks for your analytical process. And number 3, always define your terms before using them and critically check those data limitations.
But how do you know exactly which framework the examiner wants just by reading the first word of the question? Think about it. You could know the method mark strategy flawlessly. You could know every single detail about prophase, metaphase, anaphase, and telophase. But everything we learned today completely fails if you misinterpret the hidden command words that dictate the mark band. So, how do you figure out what the examiner wants right from the jump?
To solve that, the next explainer in this series is absolutely mandatory. We're going to decode those exact command words that decide your mark band. Subscribe to our YouTube channel, Ace the Exams, at acetheexams_IGCSE to excel in your Cambridge examinations. Practice those past paper questions with the method mark strategy, and I will see you in the next masterclass to crack those command words.